Hello, everyone, and welcome to today's live broadcast, Interactions Between the Microbiome and Bacteriome, Impact on Health and Disease, presented by Mahmoud Ghannoum, PhD, and Shuchen Tang, PhD. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Zymo Research. Zymo Research Corporation, based in Irvine, California, has been providing the scientific community with comprehensive nucleic acid solutions for DNA and RNA research and epigenetics for more than 2 decades. The company's unique achievements include development of the first available microelution spin column technology for nucleic acid purification. In 2016, Zymo Research released their Zymo-Biomics line of products to help optimize workflows for microbiomics and metagenomics analysis. Their innovative mock microbial community standards were developed to validate microbiomics workflows to ensure accurate community profiling. To date, Zymo maintains the most comprehensive products and services for microbiome analysis. To learn more about Zymo Research and Zymo Biomics, please visit www.zymoresearch.com/Zymo Biomics. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. And also, please notice you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or you can use the Q&A button to let us know you're having a problem. Thank you. Now I'd like to introduce the presenters. Dr. Ghannoum received his MSc in Medicinal Chemistry and PhD in Microbial Physiology from the University of Technology in England and an MBA from the Weatherhead School of Management at Case Western. He is a tenured professor and director of the Center for Medical Mycology at Case Western Reserve University and University Hospital's Case Medical Center, UH. There he established a multidisciplinary center of excellence that combines basic and translational research, investigating microbes from the test tube to the bedside. He is also a fellow of the Infectious Disease Society of America and past president of the Medical Mycological Society of the Americas. In 2016, Dr. Ghannoum received the Rhoda Benham Award presented for his continuous, outstanding, and meritorious contributions to medical mycology from the Medical Mycological Society of the Americas. He also received the Freedom to Discover Award from Bristol-Myers Squibb for his work on microbial biofilms. Dr. Tang is a research scientist at Zymo Research. He received his PhD in the Department of Chemical Engineering and Applied Chemistry from the University of Toronto. Since joining Zymo Research in 2014, he has since been instrumental in the research and development of Zymo's microbiomics and metagenomics programs. His past experiences include work in the fields of environmental microbiology and biological process engineering, with an emphasis on bioinformatics, metagenomics, and anaerobic cultivation. Most recently, Dr. Tang has been involved in the development of Zymo Research's microbiomics portfolio. He has multiple publications in different journals, including Frontiers in Microbiology, Applied and Environmental Microbiology, and Philosophical Transactions of the Royal Society of London, Series B, Biological Science. Please join me in welcoming Dr. Ghannoum. I will now turn the presentation over to him.
Thank you very much for the introduction. I really appreciate it. So as you heard, I'm going to start by talking about interaction between the microbiome and bacteriome and their impact on health and disease. So as you know, the human microbiome now is defined as the microbial communities that live in different parts of our body, the oral cavity, the GI, and skin as well. And really, data is coming in sort of a daily fashion where we are starting to see that the microbiome could affect the health and disease of humans. Now, up until recently, most of the studies that have looked into the microbiome have focused on the bacteria and the fungal, as well as to some extent the viral or virome, was ignored. Now, I would like to change this by telling you a story, which is a personal story. When I was in England doing my PhD, I talked to my supervisor and he gave me a paper where really it showed that a rabbit who is treated with an antifungal, with an antibacterial, suddenly they start to developing fungal infections. So you disrupt the bacteria, you are going to have a problem with your fungi. And as a result, it became clear to me that fungi not only can impact our health, but also can live with bacteria, and both of them could really influence each other. And based on that, I thought that it would be very important not only to look at bacteria, but also to look at fungi, because, as I mentioned, when one is disturbed, the other really take over and start causing issues. And as you see, this happened about 43 years ago. I'm getting old by the day, my friends. So, in 2010, what I did, I wrote an article to ASM Microbe saying that while the data on the bacteriome is great, we really need to also look at the fungi. However, not much really happened after that, and of course, I don't give up easily. In 2016, I wrote an article to scientists where I showed that the microbiome is an ignored kingdom, or the fungal kingdom is an ignored kingdom when it comes to the microbiome studies. And because of this, I really suggested and recommended that we should not only look at the bacteria, but also look at the fungi and other communities living in our body, because this will allow us to have a better understanding of our health and disease. So in the same article, I looked at the number of published articles that mention the fungi or the microbiome and the mycobiome, and really before 2010, when we first published the paper, really the microbiome was not mentioned. We named it. And if you look at the slide, at the histogram here in 2015, there was about 800 articles on the microbiome. 737 of them were on bacteria, 31 on fungi, and only 12 mentioned fungus at all. So by far the vast majority is on the bacteria. So the good news is that this started to change, and if you look at the moment into the number of papers that have fungus or the microbiome, it's nearly 100 different articles. So there is a great interest in this area. And as you can see in the slides, the human microbiome or the fungal community have been studied in the oral cavity, lungs, GI, as well as the skin. And in this slide, I colored some organisms or some fungus with red because these are known to cause problems and infections in mammals. So a couple of examples: Candida, which everybody knows about it. It's really the fourth most common nosocomial bloodstream infection. We have it living in our body as a colonizer till the balance, as I mentioned, is disrupted. Also, Aspergillus is another organism which could cause disease. This brings me to the next slide, where I would like to take you through how we started in the journey to study the microbiome. The first thing we did, we wanted to look at profiling the fungi in the healthy mouth. I had funding from the NIDCR, National Institute of Dental and Craniofacial Research, to look at the oral complications of HIV, and I thought before I do that, let me look at the oral cavity. And in that study, we looked at 20 healthy subjects, of course, obtained samples from them after informed consent, IRB approval. The demographics, they are all adults, 8 females, 12 males, and the inclusion criteria, they had to be non-smokers, non-diabetic, and they did not use any antimicrobials for 3 months. We also conducted an oral exam to make sure that they have healthy oral cavity or healthy mouth. We collected what you call oral wash, 15 ml in saline, and that's what we use to extract the DNA and start characterizing what organisms are there. And this slide shows you the overall distribution of fungi in the oral rinse samples from the 20 healthy subjects which we talked about, and you can see a nice heat map there. But in summary, we detected 74 culturable and 11 non-culturable genera. And the total species was 101 fungal species were isolated from these 20 people. Now, if you take one individual, you see between 9 and 23 different species were isolated from different people. At the same time, we wanted to look at really what is the core microbiome, because as you know, when we are eating, we could introduce transient organisms. And to try to eliminate those, we looked at what are the organisms that are present in at least 20% of the participants. And with that, we found 15 different genera were present, and we called it the core microbiome. And these are represented in this histogram, where, as you expect, you have non-culturable, similar to what we see in bacteria, a number of organisms that you cannot culture. Of course, Candida was by far the most prevalent, and it was present and isolated from 75% of participants. We have Cladosporium, and this organism really is not a surprise, because Cladosporium is one of the allergic fungi, or one of the fungi that can cause allergy, and it's present everywhere. But again, we saw Aspergillus, Fusarium, and Cryptococcus in these healthy subjects. Okay, and these organisms, as you know, can cause infection, but they are living there quite happily when you are healthy. Now, that paper, it was published in 2010, and it was reviewed by Dr. Arturo Casadeval, where he talked about it in the Faculty 1000, where he mentioned that there are surprises. In addition to what we expect, which is Candida and Saccharomyces, we really saw the unexpected, such as Aspergillus, Fusarium, and Cryptococcus. Also, 1/3 of the fungi were unculturable. The other thing which you see in the bacteria as well, there is very great variation, individual variation in the fungal flora as well among these individuals. And really the importance of this study was the fact that it provides us a better insight into the complexity of the human oral microbiota in general, and it shows that it's not only bacteria but also fungi as well, and of course viruses, archaea as well are part of that microbial community. And this also allowed us to start thinking, okay, this is in disease, this is in health. What happens when you have a disease state? What happens to the fungi there? And here I want to talk to you about really the microbiome, a tale of two diseases. I'm going to talk about HIV as well as the Crohn's disease. So first we'll start with the microbiome and HIV infection. As I mentioned, we looked at the healthy microbiome in the mouth, and then we wanted to see, are there changes in the oral cavity when a person has HIV disease? And to get an answer for that, we recruited subjects, 12 uninfected and 12 HIV infected individuals following informed consent were recruited. The uninfected controls were from students and staff at Case Western Reserve University, and the HIV infected subjects were recruited from the Oral HIV AIDS Research Alliance as well as the AIDS Research Unit at Case Western Reserve University. We got information. For example, we got their antiretroviral therapy, viral load, CD4, smoking status. All of these were collected, and they were all adults who were involved in this study. We had similar exclusion criteria, no antimicrobial or both topical as well as systemic, steroids, pregnancy, and insulin-dependent diabetes were excluded, subjects with that were excluded from the study. Now, unlike the first study, we only looked at the microbiome, the fungal community in health. In this study, we said, you know what, we should also look at the bacteria. Even though I am biased and I think fungi is everything, it's not the case. So we thought it's better if we look at both the bacteria as well as the fungi in the same samples. And this is the data for the oral bacteriome of the HIV-infected and uninfected individuals. This heat map shows you what we saw, but really to make it simpler, we also looked at really what are the organisms that are elevated in uninfected people or healthy people versus those with HIV infection. And this slide shows you histogram in blue, the uninfected, and red in HIV infected. And you can see Streptococcus by far is the most abundant in both groups. And you see sometimes Serratia, for example, in blue is only present in the uninfected. You can see Fusobacterium, Porphyromonas gingivalis. So it's really the usual subject which you subjects which you see in these patients on the oral cavity of healthy as well as now in HIV infected patients. So in general, we found between 9 to 14 taxa in the HIV infected, and they were predominantly present the Provitella, Streptococcus, and Rothia, whereas in the uninfected, again, similar number of taxa in the oral cavity, but we had Fusobacterium, Provitella, as well as Streptococcus in the uninfected population. Now, when you look at, we put in this Venn diagram to try to show you what sort of organisms we found, bacteria. And as you can see, in total, we found 14 genera, which constituted the core oral bacteriome. Is defined as we defined it before. It wasn't present in at least 20% of the people. And you can see the vast majority of organisms were common between the two groups. We found only in the uninfected was Agrigobacter present only there, whereas in the HIV infected, Capnocytophagia was only present there. But really the vast majority were common organisms, and this data suggested to us that we don't see a lot of shift in the bacteriome in HIV infected patients when we look at the oral cavity. Now, this is the slide, heat map again, but this time I'm presenting to you the oral microbiome or the fungal community in both HIV infected and uninfected. And you can see how it looks. But if you go closely at this histogram in the same way, blue elevated in uninfected and red elevated in HIV infected. And you can see very clearly that Candida is highly elevated in HIV infected patient, whereas the next one to it, Pichia, it's only present in the blue, which is the uninfected population. And you can see we start see differences as well, where you see more Epicoccum, for example, elevated in HIV infected people. So next slide. It shows you again similar Venn diagram, but this time we find what fungus is common between the two genera. The Candida and Penicillium is common between healthy as well as HIV infected, whereas when you look at the uninfected, we had Pichia, Cladosporium, and Fusarium was elevated there, whereas in HIV infected Alternaria, Epicoccum, and Trichosporum. Okay, when we looked specifically at Candida, it's what we expect, that Candida albicans was the most common, and we are present in 58% in uninfected and 83% in HIV infected, and also Candida dubliniensis present in 17% in both groups. And as you probably know, Candida dubliniensis, a lot of the time in HIV infected patients, when they develop oral candidiasis dubliniensis is one of the species in addition to Candida albicans that can cause this disease. So however, this figure shows us that unlike the bacteria, we see more differences in the fungi when we look at disease versus healthy people. Next slide is summary of what we did in system biology approach in HIV infected patient. As I told you, we looked at the healthy one, healthy oral cavity. We looked at an HIV, and I showed it to you. But also, we published a paper, which I'm not going to talk much about it because of the time. We looked at the microbial metabolites interaction in HIV setting, and this we published in 2015 in PLOS Pathogen, if you like to look at it. So the other thing which we looked at is we know Candida can cause oral candidiasis. So we wanted to see what is the correlation. Is there something which correlates with the presence of this Candida, whether it's negatively or positively correlated? When we looked, first of all, we looked at Pichia and Cladosporium were detected in uninfected individuals, whereas Penicillium was detected in both HIV as well as uninfected individuals to the same extent. We found 25% in that group. Now, when we looked at Pichia, and Pichia is in the black in the histogram, we found that when you have Pichia, you don't have much Candida, and when you have Candida, which is the hatch histogram, not much Pichia. This suggested to us that Pichia is an antagonistic organism when it comes to Candida. So to test this hypothesis, our hypothesis was Pichia inhibits Candida growth, and to test this, we really wanted to evaluate the effect of Pichia on Candida. We have a huge collection of organisms in our center, so we took some Candida albicans and we took Pichia, and we start to see is there an effect of Pichia in the growth, germination, biofilm formation, which, as you know, germination and biofilm formation are important virulence factors for Candida, and we use the standard methodologies which we have in our lab. So next slide, it start to show you some of the data. This is a growth curve. What we took a spent medium of Pichia, we grew it, we took the spent medium, and we saw whether it inhibits the growth of Candida, Aspergillus, and Fusarium. And as you can see clearly, the black curve is a control. They're growing very well. When you add the Pichia supernatant, by the way, it is red, which shows that Pichia supernatant inhibit the growth of these fungi. This is the second test. We looked at the ability of Candida to form hyphae or filaments and or germination, and to see whether Pichia can inhibit that. And you can see that in the untreated you have very nice hyphae, whereas when you have PSM treated or Pichia supernatant treated, we have stunted growth of hyphae. So really it inhibited the ability of Candida to form hyphae. The next slide, we looked at activity of Pichia supernatant on fungal biofilm, and this is confocal microscopy. You can see untreated, Penicillium treated, and Pichia, and in it you will see that when we untreated, very nice biofilm, thick. Penicillium treated, which is the control, again there was very good growth, and the Pichia we have really very minimal growth of biofilm. And also when we measured the thickness in the histogram, you can see adding the Pichia supernatant made the biofilm very, very thin relative, significantly lower than untreated and Penicillium treated. Now, we wanted to take this step further into the animal model, because this is really what at the end, when the rubber hit the road, is we have a developed oral candidiasis model, and we can infect Candida and cause oral candidiasis in the tongue of mice. And we had 3 groups to treatment with Pichia, treatment with topical nystatin, which is the standard of care used widely to treat oral candidiasis by a lot of dentists, as well as we have untreated and vehicle treated control, when we had then treated for 7 days, and we did evaluation after that of different endpoints. This is the slide showing you the endpoint, where you look at the efficacy of oral candidiasis in this murine model. And first of all, the infection score. As you can see, the PSM treated had the lowest infection score, also had the lowest the fungal burden compared to nystatin and the other two control groups. And when you look in histo, a picture is worth 1,000 words. You can see untreated very a large number of hyphae forming in the tongue. Nystatin treated also in the right hand panel. Also, you see a lot of fungi, but very few PSM treated animals had fungi, really few elements or fungal elements in the tongue. So this showed clearly that PICIA can treat oral candidiasis. And with that, we were able to really get funding at the moment, and we are looking to characterize what is the component that inhibit Candida and other fungus. Now, I want to shift gear and start talking about the microbiome and Crohn's disease. And here we, relationship of Candida with the GI tract have been really well established. I'm not going to go into great detail. Since 1800, 18th century, we had data to show that oral candidiasis can extend to the stomach and intestine. So this is well known, and also some other studies in the 19th century show that it goes to the stomach, the colon, as well as the ileum. So the first, now we move into the 20th century, people start looking at the microbiome and the GI tract in health, and what they found that two studies, the first just characterizing healthy people, one just healthy people, the other looking at the effect of diet. And as you can see in blue, clearly there are different members of fungi or genera of fungus present in the GI tract in both studies. Now, what about microbiome and Crohn's disease? As you know, the Crohn's disease has many factors. It's a complex disease, and multitude of factors can mediate it. It could be genetical, it could be dysregulation of the immune response, as well as the intestinal microbiota, and seems to play a role in modulating CD-associated inflammatory processes. Now, the first paper to show that there is a relation between Crohn's disease and the microbiome was from Eliv et al., and they looked at a mouse model, which is DDS or dextrose sodium sulfate induced colitis, and they looked at the fungal community, and lo and behold, they found that there are 10 different species and they were of fungi present in these animals, and they were dominated by Candida tropicalis, 65% presence in those animals. And they concluded their study that fungi and not bacteria is responsible for aggravating the severity and inflammation of irritable bowel syndrome irritable bowel disease, rather than a cause of these symptoms. So, giving you the first indication that the microbiome play a role in colitis. Then we came into this picture ourselves, and we looked at patients. Instead of looking at Crohn's disease patients and unrelated control, what we did, we looked within families, multiplex of families where we have Crohn's disease patients and relative living with them that have no Crohn's. Also, we looked at families who are not related, but they live in the same region and they are healthy. And that is very important point with respect to the control, because you need to look at at least minimize the effect of genetics and the diet. Now, this is the PCA, the clustering analysis, and you can see the bacteriome. There isn't really blue is the relative no disease, red is with the CD in the same family, and NCU, which is the green, are not, they're living in the same area as I mentioned, but they are healthy people. Really, the striking thing is when you look at the microbiome in particular, you will see the blue and the red are very close and clustered together. Okay, whereas those from the not unrelated controls, healthy controls, they were really spread in green, as you can see. When you look at the heat map, abundance of bacteria and fungal phyla in these participants, you can see the bacteriome, again, is the usual suspect, Firmicutes. You have Fusobacterium, and you have, of course, Bacteroides as well. When we look at the fungi, you have the phyla. You can see the green. A lot of it is unidentified, but you have Basidiomycota, Ascomycota are really very well represented there. Now, when you look at the richness of the microbiome, and we looked at both bacteriome and microbiome, what I really want to tell you, to stress here, if you look at CD, which is the left side, and then NCR, they're family related, and then NCU, family healthy, unrelated group. And you can see the CD and their relatives who are healthy very close together compared to the other control group. And that is true at the family, genus, and species taxonomic level. The same is true with the microbiome. And that's why to us it's very important to look at Crohn's disease and members of their family who really are related and living together, and to see if there is difference in the microbiome and bacteriome in these patients. And I think this is a good control from our point of view. Now, we looked at the abundance of bacterial taxa. This is not much different from what other people found. Like, for example, Fecalobacterium proteniceae is known to be increased in the healthy people, and we see that's true in the relative with no disease. Also, when you look at Propetella, again, it's high up, and Ocelospira. This data, other people have shown that. When you look at the fungal abundance, and you can see there is a trend in favor of Saccharomyces being higher in the healthy relatives compared to the diseased one. And the same is true with Saccharomyces cerevisiae. When you look at the Candida, if you put all Candida species together, there is no difference between them. And lo and behold, when you looked at Candida tropicalis, we found there is an increase in Candida tropicalis in this patient population. Now, abundance of Candida species, these are the different species, and you can see of Candida, you can see only Candida tropicalis was significantly higher in the CD patients. The next slide shows you the anti-Saccharomyces cerevisiae antibodies. This is a diagnostic marker for Crohn's disease, and in it you can see in CD patients it was very high, whereas in the relative healthy as well as the other healthy control, it's very low. But really what's important is that ASCA level was correlated with the abundance of Candida tropicalis. Next slide. This is too complicated of a slide. I'm not going to go over it. It just tells you what we do after this. We looked at bacterial, fungal, and fungal correlation to see are they related together. If it is red color, the circle, this means it's negative association, whereas blue, it's positive association. When we looked at the fungal, the same sort of analysis, I just want to draw your attention to the Saccharomyces, which you can see the rectangle, the horizontal one, and when you look at the Candida, the other line, you can see red spot indicating that there is antagonistic interaction between Saccharomyces and Candida. Whereas when you look at the healthy people, NCR, there is no antagonism there. Now, we looked also, since Candida tropicalis was elevated, what sort of bacteria are correlated, and we found 13 different bacteria there. And here we focused mainly in E. coli and Serratia marcescens, because they are not only correlated to Candida tropicalis, but also they were increased in CD patients. And we hypothesized that Candida tropicalis, EC, and SM, they cooperate in CD to influence host immune response, particularly in the setting of biofilms, because as you know, organisms in our gut are not just free-floating, they form biofilms and they live inside it. So what we did, we tried to see what happens when you put these organisms together. For example, alone, Candida tropicalis alone, you add E. coli, you add SM, and then you put the three together. And you can see the stark difference between when C. tropicalis alone, there is no hyphae. When you add the other organisms, we start to see a lot of hyphae forming. And remember, this is a virulence factor for Candida. We also looked at the thickness of the biofilm, and you can see in the histogram as well as confocal microscopy in nice colors. When you put the three organisms together in black, the thickness of the biofilm is significantly increased, telling you that these organisms, they come and work and play together. This is some scanning electron microscopy just to drive the message home. Again, CT alone is yeast. We'll start to see hyphae when you add the other organisms. But also this slide, I want to show you Candida tropicalis and E. coli. E. coli is very intimately, it's as if it's fused to the yeast. It's really very intimate contact. We also, I think this is the same. I'm going to skip this one, take you to this transmission electron microscopy picture, and you can see down in C, you have a big yeast. And look at the bacteria lining themselves around it, and you can see some of them are really close together. And when you look at panel D, you can see that really some here I'm trying to see here you can see some filament which really from this one is SM, EC and CT, as if the Serratia marcescens is binding the three together. Okay. Now, in the next slide that's what we came out with the working hypothesis, is that in healthy microbiome and bacteriome, you have the epithelial cells healthy, you have the mucin, and in it you have the Saccharomyces cerevisiae, less Candida tropicalis, you have more like Mycobacterium praesense in red, you have also other Oscillospira increased in these. These organisms have been shown to really reduce the inflammatory marker, pro-inflammatory markers, and we have homeostasis in this setting. When you come to the disease state, you can see in the left-hand side how bacteria and fungi, they form thick biofilms and they produce manoproteins, aspartic proteinases, and these what they do, they really have been shown to increase pro-inflammatory cytokines. Also, sometimes you have a mucolytic enzyme which they break down the mucin, and that's what caused an increase in pro-inflammatory cytokines. And the one in the right is really magnification, showing you how Candida just goes through the intestinal epithelial set. Now, this was published in MBio, and to my delight, it received a very, very big reception from media, and really it made people aware that we really need to look at fungi and bacteria, not only bacteria alone. Okay, and then I wrote another article following that in MBio. It's a commentary, cooperative evolutionary strategy between the bacteriome and microbiome, and in that article I came out with really this suggestion that microbial communities, in their yearning to survive with the host, they develop cooperation with evolutionary strategy to help them to live within the robust biofilm. Fungi will gain through that by having filamentation, increase in virulence factor. Bacteria is protected against antimicrobials, and also they secrete some mucolytic enzyme which can degrade the protective mucin layer and allow the organisms to invade and cause inflammatory responses, and the host unfortunately is negatively impacted by this process. So this figure was in my commentary, and what's important about this, it's not only derived based on my study or my group study. Also, in one study by Kellan et al. Shows that in wound you have biofilms with both covered by biofilm matrix, bacteria and fungi, and really increase inflammatory responses. And if you want to treat them, we need to start thinking of new ways of disrupting this biofilm, whether using antifungal, probiotics, antibiotics, or enzymes, and that's really how the thinking should start from now on. Now, collaborators, this is the people who helped me in the study at our Center for Medical Mycology, as well as Division of Gastroenterology and Liver Diseases from Case Western, my collaborator from INSERM at University of Lille in France, Danny Pona, Wallams indeed, as well as in HIV work with Michael Lederman and Nick van der Berg, and this is the funding. And that's the last slide. I'm not going to take much time about it, but this is my son. He goes from BMX biking to skydiving, and my heart is really rate is increasing whenever I see him. Thank you very much.
Thank you, Dr. Ghannoum, for that informative presentation. We will now hear from Dr. Tang.
Hello, everyone. This is Sichuan Tang from Zymo Research. I'm speaking here from Irvine, California. So I'm a research scientist at Zymo Research. So today, I will talk about the Zymo Microbial Community Standard and give some examples of its application to address bias in microbiomic research. So this is the outline of my presentation. At the beginning, I will talk about the need for standardization in microbiomic research. Then I will introduce the Zymo Microbial Community Standard and share some of our experience in applying the standard to address bias in microbiomic workflows. In the end, I would like to take this opportunity to talk about addressing bias during sample collection and preservation. So as you know, the field of microbiomics has grown rapidly in recent years. However, this new field has been criticized for poor data quality and especially poor data reproducibility. This slide shows an example to illustrate the problem. This is a story adopted from Science News of 2014, and the author divided one stool sample into two and submit each one of them to two well-known organizations for gut microbiome profiling, American Gut and uBiome. But surprisingly, the two profiles coming back were dramatically different. They both agree that the community is dominated by bacteroides and firmicutes. However, one shows about 33% of firmicutes, and the other shows about more than 60% of firmicutes. With one identical single sample, two well-known microbiome profiling organizations give different interpretations, and this is the problem. The next example is even more striking. Human Microbiome project and the project of metagenomics of the human intestinal tract MetaHit are the two well-known microbiome sequencing projects ever conducted. However, their interpretation of human gut microbiota is quite different. This figure illustrates the average human gut microbial profiles measured by the two projects. The data is derived from shotgun metagenomic sequencing. This figure is summarized from some analysis data downloaded from the website of Metaphyllum 2, a program come from Dr. Curtis Hartenshauer's lab. And as you can see, the abundance of the bacteroides and firmicutes is dramatically different between these two profiles. As the HMP samples were mostly connected from the US population and the MetaHit samples were mostly connected from the European population, does this mean that the gut microbiota of these two populations are so different? Unfortunately, this might not be true. A paper published in 2014 suggests that these differences are likely technical rather than biological. In this paper, the author compares the DNA extraction methods from the HMP and the meta-hit using one single stool sample. And one conclusion from the paper is that the HMP DNA extraction protocols extract the DNA of bacteroides more efficiently. Unfortunately, with their data, it's uncertain which DNA extraction protocol is more accurate because the real microbial composition of the stool sample used is unknown. The problem of poor data reproducibility across labs should not be too surprising because microbiomic workflows are complicated. They have multiple steps, and researchers face multiple options in each step. Many protocols or kits widely used in this field were developed before the takeoff of microbiomic research. Therefore, without considering the specific requirement of microbiomic research, which we believe include, for example, being unbiased and of low bioburden. This slide illustrates various factors in each step that can easily introduce bias into the workflow. So Thermo Research is not the only organization that recognizes the need for standardizing microbiomic workflows. National Institute of Standards and Technology is making a reference material currently for microbiome measurements. And there are organizations like International Human Microbiome Standards, Microbiome Quality Control MPQC Project, and etc. This is a paper published in 2015 titled An Assessment of U.S. Microbiome Research, and it identifies standardized protocols as a common need for this field. This is a paper published by the MPQC project, and it says, addressing the sources of variation in microbiota profiling is critical for optimizing protocols. Unfortunately, variation at each step in the pipeline is enormous, from physical specimen collection and processing to computational quantification of microbiome communities. So in order to address the bias in the microbiomic workflow, the use of reference material is necessary. Thermobiomic Microbial Community Standard is a product that we released last year. It is an answer to the urgent need for reference material for microbiomics measurement. So basically, this is a mock microbial community consisting of 3 gram-negative bacteria, 5 gram-positive bacteria, and 2 yeast strains, representing different degree of cell wall recalcitrance and different cell size. The microbes included covered a wide range of GC content, and it is available as both cellular and DNA format. This standard can be used as a defined input for microbiome profiling, assessing bias and errors in the workflow. The creation of the microbial community standard starts from single colonies from agar plates. Each individual organism is cultivated separately, quantified by both cell counting and DNA quantification. After that, microbes were pulled together based on a predefined composition. After pooling, the microbial composition of the standards was confirmed by sequencing-based approaches. The DNA standard is created in a similar but independent way. After pooling the pure cultures together, we further validate the microbial composition using the NGS-based sequencing techniques. These 2 figures show the microbial composition of the standards determined using shotgun and stickiness sequencing. The results show that both the microbial and the DNA formats of the standards have a composition that agrees very well with the predefined composition. An important feature of a good reference material is to have a low impurity level. We sequence the standards by high-C sequencing of 178 million reads, but only found 1 potential contaminant from Proteobacterium acnes, a common skin organism. we guarantee the impurity level lower than 0.01% by DNA abundance for all our standards. This is a powerful threshold because when using the standard to assess your microbiomic workflow, as soon as you see foreign taxon with an abundance higher than 0.01%, you can conclude that it is not from the standard and must be introduced somehow by your workflow. And this allows you to identify and control noise or false positives introduced in your workflow. So next, I will describe some examples of how we use the thermobiomic microbial community standard to reduce bias in microbiome measurement. So first, let's look at DNA extraction. So bias in the microbiome profiling caused by the use of different DNA extraction protocols has been reported a lot in the literature. And we believe this is the major source that caused the problems of poor data reproducibility across labs. This figure shows 4 dramatically different profiles of one single stool sample when sequenced and analyzed using 4 different DNA extraction methods. The first one is our product called thermobiomic DNA mini kit, and the other 3 are the 3 most cited methods currently used in this field, including the Human Microbiome fecal DNA extraction protocol, the Power Soil kit from supplier M, and a fecal DNA extraction kit from supplier Q. But unfortunately, such a comparison won't tell you which one is better because the real composition of the stool sample is unknown. And this is exactly why we need a reference material for microbiome measurement. This figure shows the same comparison but using the thermobiomic microbial community standard as the input. Unlike previous comparison, this time we know the real composition of the input. And as you can see, Thermos kit is the one that agrees with the theoretical composition, and the other 3 bias dramatically. They all have difficulty recovering DNA from top-to-light organisms, especially the 3 toughest ones included in the standard, the Listeria monocytogen, Staphylococcus aureus, and Enterococcus faecalis, all of which are gram-positive bacteria. On the other hand, they overestimate the abundance of easy-to-lyse microbes, such as Salmonella, E. coli, and Pseudomonas, which are gram-negative bacteria. So now let's go back and look at this figure again. When compared to the profile generated using the thermobiomic DNA mini kit, the other 3 protocols have overrepresentation for Proteobacterium and Bacteroides, and underrepresentation for Firmicutes and Actinobacterium. Coincidentally, most Proteobacterium and Bacteroides are gram-negative, easier to lyse, and Firmicutes and Actinobacterium are mostly gram-positive, tough to lyse. This again indicates that the other 3 DNA extraction protocols bias towards easy-to-lyse microbes. The yield data on the top shows that our DNA extraction kit has much higher efficiency than for both cell lysis and DNA extraction, while others are only scratching the surface. What causes the differences? The difference stems from the different cell lysis mechanisms. Our DNA extraction kit is solely based on mechanical lysis with speed beating. The Supplier Q method is solely based on chemical lysis, which is well known to have difficulty in lysing gram-positive bacteria. Supplier M is a combination of chemical lysis and mechanical lysis. The HMP protocol is the combination of thermal, chemical, and mechanical lysis, but unfortunately, combining more cell lysis mechanisms does not equal to less bias. Next, I will share some of our experience in optimizing the 16S library preparation kit using the DNA standard. 16S gene-targeted sequencing is the first choice of many researchers considering profiling the bacterial proportion of a microbial community because of its robustness and low cost. The major challenge in library preparation process for thickness sequencing is the formation of PCR chimeric sequences, which are resulted from the recombination of different PCR templates. These sequences are totally artificial and should be removed. Using the zymobiomic microbial community DNA standard, the extent of PCR chimera can be assessed accurately. In the PCR reaction shown in this figure, 30 cycles can cause PCR chimeric sequences to occupy more than 35% of all PCR products. And the best way to control PCR chimera is to limit PCR cycles. However, without a good solution, we sometimes have to choose convenience over data quality. And this is probably why the HMP thickness protocol recommends 30 PCR cycles, and the EMPDS Microbiome Project thickness protocol recommends 35 cycles. We will soon release a new thickness library preparation kit to help address this challenge. And one thing we do differently is to move the thickness library preparation from regular PCR to real-time PCR so that it is possible to monitor and control the progress of each PCR reaction. Besides PCR chimera, the sources of artifacts in thickness sequencing include reagent and process contamination, sample cross-contamination, barcode contamination, MySeq run carryover, PCR errors, and sequencing errors, etc. The well-defined composition and the extremely low impurity level of the zymobiomic microbial standard allow us to capture these artifacts in thickness sequencing. The pie chart on the left-hand side represents the genus level microbial composition of the zymobiomic microbial standard, sequenced and analyzed with a regular workflow, in which no measures were in place to control these artifacts. The profile indicates the presence of as many as 43 bacterial genera, while the standard should only contain 8 bacterial genera. The pie chart on the right-hand side is the corresponding profile determined using our optimized workflow, a workflow that includes procedures to ensure low bioburden, low PCR chimera rate, well-conducted MySeq run, and sophisticated bioinformatic analysis. Now the pie chart only have 8 bacterial genera, just as the standard should have. Yeah, sorry. Okay, the next one. So the zymobiomic microbial community standard can be used to assess different shotgun library preparation methods as well. So using the zymobiomic microbial DNA standard as an input, one can compare different library preparation methods. So here we compare 2 library preparation methods for shotgun sequencing, an internal method and the Nextera XT kit, a popular kit provided by Illumina. While the profiles from our internal method agrees with the theoretical value, the one of Nextera XT kit shows some disagreements. The biggest difference comes from the organism of Staphylococcus aureus. The organism happens to be the organism that has the lowest GC content in the standard. The zymobiomic standard contains organisms that span a wide range of GC content from 15% to 85%, which allow us to thoroughly assess the effect of GC variation. By plotting the sequencing coverage against GC content, one can easily examine the effect of GC content variation. And we found that Nextera XT kit has underrepresentation in both low GC and high GC content regions. The problem appears more severe in low GC content region. And which explains why we saw most bias in the estimation of the abundance of Staphylococcus aureus, as shown in the previous slide. So in the end, I would like to talk about addressing bias in sample connection and preservation. As the first step of the whole workflow, sample connection and preservation is critical to the success of the whole microbiomic project. This figure shows how microbial composition of a stool sample can change when stored at room temperature in the course of 1 month. Actually, the composition has shifted dramatically in just about 1 day. One clear trend we've seen here is that the abundance of Proteobacterium increased dramatically. And we think this makes sense because many Proteobacterium, including E. coli, are facultative anaerobes, and their growth can take off rapidly after exposure to oxygen. To prevent the bias in the step of sample connection, Zymo has developed a reagent called DNA RNA Shield, which when added into a sample can preserve DNA and RNA profile at ambient temperature in the course of 1 month. This figure shows that spike in DNA and RNAs in different sample types are stable over 1 month. You might ask, what about its effect on microbial composition in a real sample? Here are some data showing that the microbial composition of a stool sample that are saved in DNA RNA Shield at ambient temperature and sample over the course of 1 month. So first, we confirm that adding DNA RNA Shield itself does not introduce bias. And then we found that the microbial composition of the profile stay almost constant over the course of 1 month. So this shows the microbial composition at both phylum level and genus level in 16S sequencing. Besides great performance in preserving DNA RNA profiles, DNA RNA Shield also offers other benefits. It can inactivate potential pathogens in a sample, therefore eliminating biosafety concerns, which is important in the process of sample connection, sample transportation, or DNA RNA extraction. Samples saved in DNA RNA Shield can go directly to DNA RNA purification with no need to remove the reagent before purification, which is required by some other preservatives, such as unrelated. Such an operation of reagent removal can potentially introduce bias. So in the end, I would like to briefly summarize what this presentation is. So hopefully I've convinced you the importance of controlling data reproducibility, ensure good data reproducibility across lab, and hopefully I've demonstrated the utility of reference material in microbiome management. And Zymo is dedicated to providing accurate and robust tools to facilitate microbiome research. So in the end, I would like to thank all my colleagues at Zymo Research, especially to Dr. Larry Jha, our director, and our director in the R&D department, and Ryan Kem, our director in the DNA purification department. So I will end here, and I would like to also mention that we are Zymo will have a booth at ASM 2017, and we welcome everyone to join us and have the discussion about microbiome. Thanks.
Thank you, Dr. Tang, and thank you both for your informative presentations. It is time for Q&A. If you have a question you'd like to ask Dr. Ghannoum or Dr. Tang, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Okay, let's get started. Our first question is, how do chimeric sequences form during 16S library preparation? And Dr. Tang, that one's for you.
Oh, okay. So there's no, I think there's no common, there's no solid conclusion about how PCR chimeric sequences were formed during a PCR amplification process. One of the hypotheses is that maybe for some reason, let's say in a PCR cycle, when in a PCR cycle, the PCR products were elongated, but the elongation process was somehow terminated before it reached the end. And this partially amplified PCR product was later on used as primers for subsequent PCR amplification process. And therefore, when it binds to different PCR templates, the PCR chimeric sequences were formed. But as I mentioned, currently there is no general conclusion about how these chimeric sequences were formed, and one of the most effective way people have found is to control and limit the number of PCR cycles you run for the PCR reactions. Yep.
Dr. Tang, how does GC content variation cause bias in sequencing coverage in shotgun sequencing?
So in shotgun sequencing, that's a good question. But again, there are some hypotheses about how GC content, or there are some observation of how GC content can affect sequencing process. The first step will be maybe in the library preparation process. And as we all know, GC content could have a significant impact on the PCR amplification process. And a lot of library, next generation sequencing library preparation process uses PCR steps. And that's potentially how GC content can affect can affect library preparation process. And also there are some earlier, I remember there are some papers also have some discussion about how GC content can actually affecting the sequencing chemistry on different sequencing platforms as well.
Dr. Tang, what makes your isolation kit less biased than others?
Okay, so as I mentioned, a lot of kits or protocols people currently use in the field was designed before the takeoff of the microbiomics research. So as you know, a lot of, for example, like a lot of applications we used before is maybe you will focus more on like a qualitatively qualitatively identification of microbes. For example, like using PCR reaction to identify the presence of certain microbes. And in that context, you actually do not care how well you lyse the organism, as long as some of the organism was lysed in the sample, and using a PCR technique you can identify it. It actually doesn't matter like how well you lyse that sample. But now as I mentioned, the microbiomic research posed some new requirement about how well you should lyse the sample. And have that in mind, we designed this new DNA extraction kit, and it's to make sure all organisms will lyse evenly and completely. And the other important feature of this kit is the having very low bioburden, and that's also very important as well. And the way how we make sure it has minimum bias is there are two differences. One is the BBT matrix we use. It's a mixture of two different kinds of beads, and these beads also have of very high density, so they are very heavy and also hard. And also the other thing we do differently is in the lysis solution we have some special chemistry there to help protect the DNA from fragmentation, even during like the intensive mechanical lysis like beating process.
Another for you, Dr. Tang. Where can I find more info on Zymo Biomics?
Oh, you can find more, as I shown in the last slide of my presentation. So you can find more information by going to our website at www.zymoresearch.com/microbiomics.
Thank you. And Dr. Ghannoum, this next one is for you. Would you explain how you measure or sequence the microbiome in the human samples, and what database do you use, and what region or gene do you sequence?
Okay. So for the sequencing, we will use for fungi the internal transcribed spacer, or called ITS, and 16S for the bacteria. And with the database, we usually classification of the species level. We perform using, or what is referenced using the Unite 5.8S database, and the taxa we assign using the NBLAST method.
And Dr. Ghannoum, how do you extract to ensure you get a representative DNA sample of all fungi in the sample? And do you have any tips or tricks?
You know, it's really tough sometimes with fungi to isolate the DNA, because fungi is much more difficult than bacteria. They have very hard cell wall. In our published work, what we did is we used actually QIM DNA stool minikit to get the QIAGEN, and we followed their instructions. And it really works very nice. Sometimes you really have to do some there is some variation sometimes. Some people, if we don't get a lot of DNA, we try to break it with beads, and that should work also. But this does not happen very often.
Okay, and Dr. Ghannoum, this next one is for you too. In the Crohn study, could the differences you see between related controls and non-related controls be explained by diet? I'm wondering about gluten-free or FODMAP diets. Family members are more likely to eat the same thing.
Very good question. Very good question. And that's what we did is we selected the family. I mean, clearly their genetics are closer, and the vast majority of them, they live together. So the diet does not work well. When we looked at the other, which is not related healthy, we selected them to be from the same region, hoping that they will be at least eating, whether Middle Eastern food or French food or whatever. You know what I mean? So we tried to minimize that. But it is very important question. You need to consider that.
And the next one is for Dr. Tang. This person says, sorry, but I missed your comment about RNA later. Could you repeat it? Thank you.
Oh, so as I mentioned, for RNA preservation, the DNA shield is also applicable. So we've seen that we're able to preserve RNA profile using the DNA shield, like being able to preserve it at ambient temperature for more than 1 month. And we have different sample connection devices, for example, like designed for different sample types. For example, we have fecal connection cups. We also have swab connection kits. That was this device will pre-fill with DNA shield to provide the best convenience for a customer to use. And we also sell the DNA shield as bulk solution by itself as well.
Okay, Dr. Tang, the next one's for you. Are there specific RNA preservation media for each type of sample, blood, feces, and saliva, or is it the same?
Oh, yeah. As I mentioned, we have that basically I answered that basically also in a previous question. So basically, we have different devices that was designed to facilitate the sample connection process for different samples. Like we have the blood connection tubes, fecal connection cups, swab connection devices. And all of these will all pre-fill with DNA shield to help preserve those DNA profiles in these sample types.
Okay, thank you. And next one's for Dr. Ghannoum. Do you think that fungal influence bacteria under any conditions? Maybe that's fungi. I'm studying the tick-borne pathogens. Do you think that I have to take a look at fungi harbored by ticks and search for interactions?
this is very good question also. I think definitely, at least from more than one study, fungi and could affect the bacteria there. So in ticks, if you have again, I don't know really the background, the microbiome of ticks, but if they have fungi and bacteria in ticks, so I expect that they will cooperate together and they will influence each other. And as you know, like if we look at the plants, for example, a lot of the time, sometimes fungi secrete some agents. For example, penicillin was secreted by Penicillium chrysogenum. So definitely there are some metabolites secreted by fungi could affect the bacteria as well.
Great. I would like to once again thank Dr. Ghannoum and Dr. Tang for their presentations. Do you have any final comments?
I think this is a really great venue. I learned a lot from Dr. Tang, and I hope we also conveyed the idea that we should expand our outlook to both bacteria and fungi, and of course to use the standardized method which Dr. Tang mentioned.
Yeah, it's great honor to have a presentation here, and I also learned from Dr. Ghannoum's presentation a lot. And actually, my second time of listening to his presentation it's really intriguing.
Thank you.
Well, thank you both once again. I would also like to thank our sponsor, Zymo Research, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through November 22nd, 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.